Hello, everyone, and welcome to today's webinar, Part 1, Achieving Laboratory Operational Excellence through Abbott's Clinical Chemistry Solutions. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Abbott. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education Credits window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Marvin Berman, Manager, Scientific Services, Diagnostics Division of Abbott Laboratories. Dr. Berman is responsible for scientific support and collaboration in clinical and immunoassay diagnostics. He has extensive experience in developing and supporting both immunoassay and clinical chemistry diagnostic assays, having held positions in research and development, manufacturing, technical operations, validations, and business excellence. Christina Fabro, Marketing Manager for Latin America and Canada, will begin the presentation.
Welcome to Abbott Core Diagnostics session on achieving operational excellence in clinical chemistry. I'll start with a disclaimer. This webinar is intended as a scientific educational session to inform both laboratory and healthcare professionals of the latest researched and published data. The menu is not available in all markets. If you have any questions or require any information, please contact your local Abbott representative or add a comment in the chat. The following session will highlight Abbott's commitment to helping your laboratory achieve excellence in clinical chemistry, Sigma Strong. As this image shows and represents a combination of two things, Abbott's Six Sigma assay performance, you see the performance curve, and harmonized systems. These are platforms such as Alinity and Architect. Both these two components allow you to obtain highly accurate test results. You will hear about Sigma Strong from our speaker, Dr. Marvin Berman, who's going to illustrate the key benefits to Sigma Strong and Abbott's new approach to clinical chemistry. We will focus in on three areas: on how we're bringing assay formulation improvements and continuing to leverage innovative technologies to deliver operational excellence in these three focus areas. The first one is confidence in Six Sigma clinical chemistry results, having high performance assays and the confidence that that provides, and the contribution to operational excellence. The second area will be assay productivity. Through both assay design and system design, these components can help laboratories achieve operational excellence. And the third area will be total value of ownership improvements. So the objectives of today's session will be to look at the role of Sigma metrics in laboratory medicine, an overview of CLSI guidelines and measuring intervals, and how does innovation in assay design, analytical technologies, and instrumentation help deliver high-quality results, taking the lab to a next level in operational excellence. I will now turn it over to Dr. Marvin Berman from Scientific Affairs to continue the presentation.
Thank you, Christina. So labs need to provide reliable, high-quality results to support clinician decision-making. Abbott works to develop assays aligned to the latest CLSI guidelines, which deliver Six Sigma analytical quality to help address these challenges. So what is CLSI? CLSI is the Clinical and Lab Standardization Institute, and they are a group that develops guidelines for laboratorians and manufacturers based on input from manufacturers, laboratorians, regulatory bodies, key opinion leaders, experts in the field, and they developed these guidelines that will meet the needs of all these groups. For example, EP17 on measuring interval was just released. This guideline was developed because measuring interval had many names: calibration range, linearity range, upper limit, and so it wanted to modernize these terms as well as provide guidance in how a measuring interval should be defined. And so measuring interval is defined by the sensitivity, such as limit of blank, limit of detection, limit of quantitation, linearity, method comparison, and precision. So Abbott, as part of these CLSI teams, uses these metrics as well. So limit of blank is the value you get when you have no analyte present in the sample. Limit of detection is the value you get at the lowest concentration of analyte that you can read. And LOQ, or limit of quantitation, is the lowest concentration of analyte that can be detected based on both the bias and precision. So measuring interval. The analytical measuring interval is defined as the range in which specimen concentrations are measured within the medical and lab needs for accuracy with no dilution, concentration, or other pretreatment not part of the standard or routine measurement process, inclusive of dilutions or pretreatments that need no user intervention. The extended measuring interval, shown in purple, is the range in which concentrations are measured with appropriate accuracy by diluting the specimen before taking a measurement with the developed measurement process. And so the whole range shown below is called the reportable interval, and it's the range that includes the analytical measuring interval, or AMI, the extended measurement interval, or EMI, and extends as far down as the limit of low detection. Using these harmonized techniques, laboratorians are able to compare performance of assays across different manufacturers. So Six Sigma quality is a good measurement of performance of assays across different manufacturers and systems. It's derived by the equation in the lower right. It's your total allowable error minus your bias divided by your precision CV. So in the graph on the left, you can see in the center is your target, and any difference from the target is called the bias, and the precision around the target is your CV. And in the lower right-hand corner, that area represents defects. So sigma values range from 1 to 6. 1, a low sigma value, would represent 690,000 defects per million, versus 6 sigma represents 3.4 defects per million. So as you increase in sigma value, your defects go down. And laboratories want to be at least in the 4 to 6 range for the assays that they're running. So again, 6 sigma, what does that mean? It's, again, confidence in your 6 sigma clinical chemistry results. It's having assays thoughtfully designed and system designed to meet users' needs. And it's a total value of ownership, a partnership between Abbott and the users or laboratorians. And I'm going to talk a little bit about the first one, confidence. So how can you be sure that you can have confidence in the values that you're reporting and that the assays that you're using will meet the operational excellence that you desire? So when Abbott is designing new assays or system designs, they look at metrics to assess themselves when we build these assays. They should have quality, have customer key, expert engagement, commutability, should have critical menu items, and efficiency. So for quality, does it meet modern expectations, such as the CLSI guidelines? Are we consistent in our labeling across all our product lines? Do we meet consistent sigma levels? And when we do accuracy studies, are we using a recognized standard that laboratorians can use to verify our performance? Customer and key expert engagement is important in getting customer input on features that they want and need. Having early adopters studies where customers can give feedback on the performance of the product early in development, and scientific collaboration with key opinion leaders to know what features the assay should have, such as should it be a serum assay only or should it be a serum urine. Commutability is important. We strive to make sure that when we go from a new platform such as Architect to Alinity, that results are commutable and assays perform equivalently or better. We want to make sure assays are standardized correctly to international standards, that customers get expected values, and we also work on developing pediatric reference ranges. Menu is important. Are we providing the assays that are needed, like HbA1c, drugs of abuse, therapeutic drug monitoring, and efficiency? Do our systems have error-proof designs? Can you load the reagents while the instrument is still running? Can you have onboard materials so you don't have to stop the instrument to calibrate every time? Can the instrument fit nicely into a small lab or a big reference lab? Is it easy to have lab personnel run the instrument? Can I load higher load capacity samples on the instrument? All important features to look at when you're designing an assay or a system. So again, it's a collaboration. We use guidelines prepared by organizations such as CLSI and manufacturing, and we work to meet the needs of clinical labs by listening to both the clinical labs and following the guidelines to give a product that meets customer expectations. The Sigma metrics are important to us. Here's an example from Albumin BCG. You can see it has a precision percent CV of 1.4%. It has a bias of 4.8%, which leads to a sigma value of 6.6, which is where you want to be. And then on the next slide, we have a list of 6 of our new Alinity clinical chem assays, and you can see all of them have sigma values greater than 6. They all use a recognized standard from NIST or IRM, and we did the testing, accuracy testing at a medical decision point, and where we still get very good CVs, about 1%. So now, assay productivity through assay and system design. So the new Abbott assays are built with the customer in mind. We have optimized the kit and cartridge size to make it easier for customers to load clinical chemistry assays. We have increased onboard stability, increased calibration stability, all to allow for a higher throughput and less customer intervention. Challenges. There are many manual processes that hamper efficiency and cause increased risk of error or inconsistency. Abbott develops assays and systems utilizing innovative technologies that enable streamlined workflows, reduce hidden costs, and improve efficiency to help address some of these challenges. Abbott uses unique and proven technologies. We offer pre-analytical processes like sample interference indices to evaluate hemolytic samples, lipid samples, enteric samples, so that you can make sure that there's no potential interference. And we have clotted bubble detection to make sure the sample is aspirated correctly. As well, we have technologies such as the ICT module, smart washing, and FlexRate that I'll go into in more detail in the next slides. So Abbott offers an ICT module that can measure the electrolytes sodium, potassium, and chloride. You can get a single analyte, or you can get all 3 analyte responses in a single sample. It offers 60,000 determinations per module, CVs less than or equal to 1%, and automated maintenance. Here's some of the data from the ICT module. You can see that the performance of different tube types, lithium heparin, sodium heparin, lithium heparin serum separator tubes, and serum separator tubes. CVs are very comparable across the different tube types as well. Interference by icteric, hemolytic, and lipemic samples is very low. This means that you're going to have less issues for having to do reruns or repeating draws, and gives a more consistent turnaround time, and of course less maintenance. Abbott offers a smartwatch technology that ensures minimal sample-to-sample carryover. Abbott has established that the carryover specifications meet or exceed the bias or total allowable error requirements for each assay. These specs are derived from recognized international criteria from CLIA, NACB, RICOS. Abbott ClinChem systems also have an optimum sampling sequence feature, which automatically determines the sampling sequence to minimize smartwash so that you can do a good flow of sampling without having to do a smartwash every time. So the smartwash technology was tested across 3 instruments. It looked at low sample volumes of 15 microliters and high sample volumes of 35 microliters, and used a sample surface antigen that is known to that it could cause carryover if you don't have the correct wash system set up. And you can see carryover was less than 0.01 parts per million and passed all our criteria. So this is important because it provides confidence in results that you can ensure that you're providing the proper patient care. It means you don't need to use pipette tips and alquadding to avoid carryover contamination in samples. No need to run all immunoassays first to avoid carryover in chemistry systems, and no need for carryover evasion protocols that could lead to inconsistent turnaround times. FlexRate technology, what is that? It extends the linearity of clin chemistry enzyme assays by using 2 absorbance read time windows. In green, you have a main read window, and in orange, you have the FlexRate read time. You can see a normal sample with the green dots. It's linear across the main read time and would be measured in the main read window. The orange sample with the orange dots, you can see it's flat in the main read window, and so you wouldn't get an accurate reading of the sample, and in other systems would be rejected for out of range. But on the Abbott system, you can flex to the flex read time, and you can see in that read window, the points are linear, and you can get a reading. So what does this mean to a customer? It means you don't have to repeat that sample, try to figure out what alternate dilution to use, take time to put a new sample on. Like this, you have confidence that that sample will be read, or the machine will use the flex read time to read it, saving you time and cost. Here's some performance of our enzyme assays. It shows slopes around 1, correlations around 1. You can see the wide range of each of the assays shown. And on the left, you have correlation graphs showing the Alinity versus Architect commutability of samples across both platforms.
Dr. Marvin Berman has reviewed Abbott's commitment to operational excellence. The lab plays a critical role in the healthcare system, providing results to patients they serve. Sigma Strong provides you with these 3 benefits that Marvin has reviewed. Let's go through it again. The first is confidence in Sigma clinical chemistry results. Having a high-performing 6 Sigma assay can allow you to potentially decrease the amount of workarounds, change QC practices, and also be confident in the results that you're reporting and help you with that operational excellence. We went through the assay productivity, looking at assay design and system design, just highlighted right now. So in particular, for the system design components, technologies such as ICT, integrated chip technology for your sodium potassium chloride determinations, FlexRate for enzymatic assays, and SmartWash, reducing that sample-to-sample carryover, all contribute to achieving operational excellence. And lastly, the area of focus is that total value of ownership improvements. So this is Abbott's new approach to clinical chemistry. So Sigma Strong, our commitment to clinical chemistry is bringing that assay formulation improvement and continuing to leverage our innovative technologies. So our clinical chemistry offerings provide customers with that total value of ownership. Here we have listed 2 studies. What these particular 2 studies and cases have realized is that value of ownership through reduced quality control frequency, having high-performing Six Sigma assay change potential quality control frequency due to the performance, improved turnaround times, and simplified calibration workflows, all helping to contribute to that total value of ownership and also achieving and improving operational excellence. We will be having a part 2 of this session where Sten Westgard is going to talk a little bit more about the impact of Sigma quality on lab operations. We know that high-performing assays, Six Sigma quality assays, require only 1 control rule with 2 control levels. This can allow you to streamline and optimize your QC strategy and practices, which in turn may help you save time, material, and labor. So just to summarize, the importance of Sigma quality and the impact that it has within the laboratory and beyond the laboratory. Six Sigma can benefit the healthcare system. It can reduce or decrease errors by nature of Six Sigma performance and having assays that have high Six Sigma metrics. This in turn can provide consistent delivery of accurate test results, improve that turnaround time, and ultimately, at the end of all of this, is a patient, helping to improve patient care and also physician and patient satisfaction. Sten Westgard will now introduce some of the concepts that he will be presenting in part 2 that will go live session will be on August 11th. Sten Westgard is the Director of Client Services and Technology for Westgard Quality Control. Please join us on August 11th.
Thank you for that introduction. I'm glad to be here to be able to give you a preview of our talk on August 11th. I do want to acknowledge that I am being supported by Abbott. I'm very thankful for them to be giving me the opportunity to connect with you in the next session. One of the goals of the next session is to understand why we are using Six Sigma in quality control. Certainly we know that change is constant in the laboratory. It happens with instruments and methods and automation, but even the Westgard rules and how you do QC has to change. Now these changes aren't going to make your life worse, they're going to make your life better. You'll get better results for patients, easier QC, and actually it will become less expensive for you to run those tests. When you have Six Sigma assays, you will learn that you need very few Westgard rules, and you'll see fewer out-of-control events, you'll consume fewer supplies, run fewer controls, and your staff will spend less time chasing those out-of-control events. We'll also show you some benefits in the real world. We'll show you a case study of a laboratory that found by adopting a Six Sigma approach to QC with higher quality assays, They had 85% fewer QC failures, less time they spent working that instrument for troubleshooting and QC events, which resulted over the 9-year lifespan of that particular instrument of about $100,000 of soft labor savings. And there are also hard dollar savings that we'll discuss in the session. So I hope you'll join us on August 11th for part 2 of this series and understand and learn that Six Sigma is not a destination, not a single destination, it's a path. And you have to sustain it, not just achieve it once. We'll teach you how to calculate an analytical sigma metric, identify improvements for QC in your assay performance, and design better QC with Westgard sigma rules. So I look forward to joining you. I hope you will be with us on August 11th.
Thank you, Dr. Berman, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many questions as we have time for. All right, let's go ahead and get started. Okay, your first question is, which allowable total error, TEA, values does Abbott use to calculate their sigma values?
We don't have like a specific value. We look at a particular assay and see what is recognized as a TEA for that product. Oftentimes there's more than one, so you try to pick the most restrictive. But there are many sites available, such as CLIA, that offer guidance on the TEA to select. Thank you.
Okay, thank you. Now, what is the basic mechanism of FlexRate? So
it's really instrument software. That what we do in the instrument is we set up 2 specific read time windows. One is where we expect the majority of the samples to give a linear rate over time. And usually, I can't give a specific percentage, but usually it's like 80 to 90% of the assays will give a linear response in that range. But we also have an earlier read window such that if the reagent is consumed early, the substrate reagent is consumed early, we can read at an earlier time point because we keep all the read profiles of that sample. So we can just, instead of reading later in time, say at 10 minutes, we can read at 5 minutes and report a result without the user having to rerun a sample. So it's all controlled by the instrument software. The analysis is exactly the same. We're looking at a rate of change of the sample over time. So there's no difference in the response other than we're looking at an earlier time point in the reaction in order to make sure that the rate is linear over that time. Thank you.
Thank you. And your next question, can accuracy studies be done using the product calibrators?
Yes, they can definitely be done with a calibrator, but if you want to know how your system or assay is performing across other competitor systems or across other assays, it's better to use a recognized standard. But yeah, there's no harm in doing the calibrator, except that since the product is calibrated with that material, it should definitely be accurate against that calibrated material. So you're kind of evaluating accuracy against the same material that you use to standardize. By using an international recognized standard, if the calibration is off, you'll be able to detect that with this international material, or reference material, I should say. Thank you.
Okay, great. Thank you. All right, your next question. Can you address the difference between the limit of detection sensitivity between Abbott and other manufacturers?
Well, Abbott is following the CLSI latest guideline, and so we give a value for limit of blank, limit of detection, and limit of quantitation. So when we say limit of detection, you know how we're doing it. Some of the older products may say limit of detection, but they may be actually doing a limit of blank. So you really have to look, if they're not following the latest CLSI EP17 guidance, you need to look at which CLSI guideline they're using and how they are defining the limit of detection, because often they're running a blank sample. So their limit of detection will seem lower when in actuality you would have to compare our limit of blank against their limit of detection. So you can't always go by how they're defining limited detection. You got to dig a little deeper to compare the products. Thank you.
Okay, great, thank you. Can we measure quality control using by this formula in Six Sigma metrics? Can you please explain it?
So Sigma metrics follows the formula that you choose a TEA, you choose a CV, which is derived from some sort of precision study, such as a 20-day precision study that Abbott uses, and then you're looking at the accuracy against the target values. So you can use a QC material, but it's not as good as using, say, a calibrator, which is not as good as using a reference standard. But certainly people have calculated Sigma values using QC material. It just depends on what you're most comfortable with and which material you can get, but you're better off using a recognized standard than a QC material, because QC material could have material in the matrix that affects one particular competitor system, but not another. So QC material is not as consistent as reference material. So that's why, the lot-to-lot differences between QC material can be very pronounced. And so you got to be careful when you're using those types of materials. Thank you.
All right, thank you. Does Abbott HIL serum indices provide a quantitative result for endogenous interference?
Yes, it provides qualitative as well as a semi-quantitative value, and you're able to choose whichever one you want. You could get it in default units or in international units as well. And Abbott has a clinical brochure that we provide that lists HIL expected results on the Abbott products. Thank you.
Okay, great, thank you. Now your next question, what is the shelf life and stability of the assays?
Well, every assay is specific. So if there's a particular assay you're interested in, we could probably provide that. But in general, we try to get at least 12 months reagent stability and 12 months calibrator stability. But again, it's product specific. Thank you.
Okay, great. We have a few more questions here. Your next question, what information or direction do you provide for the assay validation process? If
you work with your Abbott representative, they'll assess your lab and what you're doing and give you a recommendation. Typically, we'll look at precision, linearity, method comparison, old versus new method. So again, it's kind of assay specific, but Abbott's more than willing to work with customers to figure out the best plan for them to use. Thank you.
Okay, great. This next question, I'm not sure if it's a complete question, but it says, how many parameter and tests per hour?
The ClinChem assays, it depends on the throughput. It's about 1,300 tests per hour. If you're running the potentiometric assays and the photometric assays, it's going to depend on your mix of assays, but generally you'll get around 900 to 1,300 tests per hour. Thank you.
Thank you, Dr. Berman. We have one last question here for you. Can you explain how the precision value was obtained for your next generation Abbott clinical chemistry assays?
We did a 20-day precision study with a minimum of 2 reagent lots. So by looking over multiple days, we can make sure that we're looking at a good, we're getting a good estimation of the precision values. So I think doing 20 days really gives you a good sense of the precision. And we do it at multiple levels of controls and panels that span the dynamic range. So looking at our labeling, you can see at a specific level, especially at medical decision points, what you can expect in terms of precision for the assay. Thank you.
Thank you, Dr. Berman. Do you have any final comments for our audience before we wrap up today?
I would just like to thank everybody for listening to today's presentation. And if you have some follow-up questions later on, we're more than happy to respond to them, either through our Abbott representative or sending question comments through the lab routes. Thank you.
Great. Thank you again, Dr. Berman, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Abbott, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Please join us on August 11th, 10 a.m. Pacific, 1 p.m. Eastern for part 2 of this series presented by Sten Westgard. Sten Westgard, Director of Client Services and Technology for Westgard Quality Control, will discuss how stronger harmonization builds better assays, simplifies QC, and strengthens results. Until next time, goodbye.